Then later your CC jalan off. Hey, what do you mean no care? I purposely invite one speaker down there to speak to you over Zoom.
Oh, I thought we going to have a confirmation again.
Yeah. I think we have to send your class a Zoom link for the speaker, since all of you can't listen to this. Yesterday,
Is it like
maybe it's just me.
yesterday, I think.
Yeah. Okay. I will do my narration. Okay, to help you visualize the page 8, this is what's going to happen. I'm just going to narrate over this video to give you an idea of what's happening. Okay, here you see a lot of thylakoids, and we're going to zoom in specifically to the thylakoid membrane. Above, on the outside of the thylakoid is stroma, inside of the thylakoid is the thylakoid space. You find a lot of proteins on this thylakoid membrane. We call this chain the electron transport chain, okay? Just like in oxidative phosphorylation. What's different, however, is that in plants, they have these two extra names, photosystems. Photosystems are nothing more than a collection of proteins that contain the very pigment that we observed yesterday, chlorophyll. There are two photosystems, photosystem 2 and photosystem 1. Right? If you want to refer to a diagram, you can refer to the diagram in your notes. Your notes will have labeled it photosystem 1 and photosystem 2. You may be wondering, why is this the second one photosystem 1 and the first one 2? That's because we discovered this one first. Then we discovered this one after. So that's why the naming convention is a little bit odd. The purple things in between, they are also proteins. Do you want to make a guess what this last large protein is? Yeah, looks the same, right? That's because it is the same. ADP synthase. The same one you find in mitochondria. You see a lot of things striking the photosystems. Think of those as the light, the photons of light from light energy, striking the photosystem. Okay, as a biologist, I don't quite know what photon exactly is. It is a particle. Made of what? Not too entirely sure. Who's your physics teacher? Miss Ho. Oh, Miss Ho gonna go already. Yeah. Okay. Don't know why I touched that. Okay. So as the light energy strikes the various chlorophyll pigments, what will happen is that the electrons in the chlorophyll pigments will get very excited. But rather than release the energy into the environment as wasted energy, the electrons instead will transfer energy. Right? And the energy that's transferred will eventually excite these electrons to the point that they will leave the photosystems altogether. Once it leaves the photosystem, it will now flow down the electron transport chain, just like in oxidative phosphorylation. So if you observe, okay, here goes the electrons again. Once it gains enough energy, it will be excited out of its photosystem. It will be transferred down the electron transport chain. But now we have a problem. This photosystem is now missing electrons. Well, it's okay, we can top it up by splitting up water. See the water molecule over there? We're about to split it. And that releases electrons, and we produce oxygen as a byproduct. Notice, isn't this kind of like a reverse of oxidative phosphorylation? In cellular respiration, oxygen was the final electron acceptor. In this case, water is the one that gives up the electrons, and then we get oxygen at the end. Really, what we are doing here is doing the reverse process. Let's watch what happens as the electrons flow down the chain. Okay, as the electrons flow down the electron transport chain, the usual stuff happens. Protons will get pumped. Except if you notice, the protons are being pumped from the outside in. It's being pumped from the stroma into the thylakoid space. Once we've set up a proton gradient, the usual stuff happens. The protons will flow through ADP, and then ADP will be used to synthesize ATP. Notice it's very similar. That's why if you understand oxidative phosphorylation, photosynthesis just becomes almost like a reverse. And at the end, remember oxygen in oxidative phosphorylation was the final electron acceptor. Same here in this electron transport chain, we also need a final electron acceptor. Right? I'm going to make a guess what will be the final electron acceptor. Right? If we think along the reverse. Hydrogen? Okay, remember who was the one donating electrons in oxidative phosphorylation? H2O? In cellular respiration. Remember NADH, right? In oxidative phosphorylation, we've been storing a lot of NADH, and then we will release electrons at the start, right? In this case, the electrons get transferred to the plant's version of NAD class, which is NADP class. Okay, the P does not stand for plant. I cannot remember what it stands for, but it's not plant. But okay, great. I think that's a better way to remember it. NAD plant. Okay. When it receives the electrons, it acts nothing more like an electron carrier. Just collect electrons again. So think of it this way, we are really going the reverse. Once it collects the electrons, it will become NADPH. So as we zoom out, let's look at the overview. Electrons get passed down the electron transport chain, and as it passes, protons keep getting pumped into the thylakoid space. And this proton gradient will help to get ATP synthase working. In other words, truly, this is really like a reverse of oxidative phosphorylation. Yeah? Right now, on the whiteboard, I'd like to show you a little bit more detail about how this process works. I have not given a name to this process. This process is called the light-dependent stage because this process requires light. I'm going to now strike light onto the photosystem. Okay? And now I'm going to simulate what happens when light strikes the photosystems. Here, every circle in the photosystem represents a chlorophyll. Every yellow magnet represents the electrons inside that can be excited. Okay, I have no better way to demonstrate this. So just bear with me. It's going to be a bit weird. When I show an excited electron, I'm going to like, okay? So if it jumps out from our plane of view, means it's excited. When it goes back down, means it is not excited anymore. Okay? So this is what happens. As light energy strikes the electrons in the chlorophyll, they get excited. And then, as it goes back down, because it's unexcited, the energy is not wasted. Instead, red iron, yes, red iron. Let me suck myself up again. As I was saying, as the light energy strikes the electrons, it gets excited. But when it goes back down to the ground state, energy is not going to be wasted. Instead, you pass it on to the next one. Have you ever been to a soccer match or some match where you do a wave in the crowd? That's essentially what they're doing here. You're not really touching each other, but what you're doing is you're just transferring that hyped up energy to the next person beside you. Yeah? So the electron's excited. As it goes down to ground state, it excites the next one. As it goes down to ground state, it excites the next one, and the next one. And now you think about it, I have so much energy being transferred to this middle electron over here. So something is going to happen. As this electron gains all the energy from all these, the electron, instead of just getting excited, it gets so excited, it will jump out of its chlorophyll into this structure at the very top. Okay, in your notes, they refer to it as a primary electron acceptor. Okay, the primary electron acceptor is just this small little structure over here. It is what will receive the electron after it jumps out of its chlorophyll. Simple, right? Yeah? The same thing happens at all the photosystems. This photosystem over here will experience the same thing. So, again, excited, 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 and as it goes back down, it's going to transfer all the energy to this electron, and it's so excited it will jump out of the chlorophyll into the primary electron acceptor here. But what's the point? The point is, we need to get it out of chlorophyll so that it can flow down the electron transport chain. Can I say again? We need to excite it to the primary electron acceptor, because from there, then it can start going down the electron transport chain that we are so familiar with. And therefore, if you were to look into your notes, you'll see this particular page, a photosystem. I'd like you to take note that the black arrows do not represent the flow of electrons. They represent the flow of energy. Flow of energy. This yellow arrow, however, represents the flow of electrons. So just take note of this small difference. Yes. You will tell us, right, if you look into your notes, you scroll to this particular page and you are very attentive, you'll see that actually 2 electrons are moving out. Okay? So I just want you to know that whatever I'm presenting up here is simplified. I do not have enough space to put 2 electrons. Okay, actually I do, maybe I put 2 like that. Okay? 2 electrons. Okay, but for simplicity's sake, I'm just putting 1 electron. Because the concept is you move more electrons. Okay, I don't need you to know whether it's 1 or 2. Okay, maybe I need the help of someone now. Aziz, will you come up for a while? Aziz will be photosystem 2, which is over here. I will be photosystem 1. Okay? And then light energy is striking. So Aziz, you do it with me, ready? Okay, you stand to the side so everyone can see. Yeah, okay, ready? So light energy strikes and then we get excited. Yeah, okay, pass it along. Okay, I'm a bit slow. Okay, jump out. Okay, let's go. Excited. Okay, pass it along. Pass it along. Okay. Now so much energy it jumps to the primary electron acceptor. Now it transfers down the electron transport chain. Okay, let's do it one more time. Ready? Go. Hey, okay, wait. Aziz, what did you just do? What you put back? It's flowing down the electron transport chain. Okay, let's go. Excite. 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 Okay, we got a problem now, right? We are out of electrons. What now? Okay, I can fix this problem for my photosystem. I'll just take Aziz one now. Okay? Okay? So my excited ones will just be transferred to this from Aziz. Okay, Aziz, where will you get your electrons from? Top up. Okay, he'll get it from water. Okay, so let's break down this water molecule, pop, release hydrogen. Now we get oxygen that the plants produce. But in doing so, electrons will come out from the water molecule into the photosystem. So that's how this electron transport chain tube tops up the electrons. It tops it up from water. Photosystem 1 tops up the electron from photosystem 2. So that's the concept here. Yeah? And I needed the help of one more person to demonstrate this. Okay, I'm going to pause now for you to take down notes or clarify anything that was not clear. Yes. We want to take up electrons from carbohydrates so that we can transfer it. Sorry, we go a little bit down first. Yes. Say again, speak up. Yes. Okay, Iman. No. It's not any. Primary electron acceptor is not NADP plus. It is, if I'm not wrong, it's a protein that is right over here. It's just another protein. Yes, Emily. Oh, okay. So just now we kind of depicted it like that, isn't it, Emily? Like we went from the primary electron acceptor, then I like that, is it? Yeah. Okay. It does not go into the stroma. What would be more accurate is that it's transferred to the protein directly. Okay. I'm also missing some proteins in between. If we watch the video again, okay, let's watch the video again. You will see that there are some in-between proteins that help to shift the electrons. Okay, I think this part will be clearer. Okay, watch out. Okay, there are some shuttle proteins that will help to transfer electrons. Yeah. Okay, let's finish this process. Where will the electron go? Right? We've extracted it out from water, goes down the chain. Now we've reached the last step. Who will accept the electrons? Okay, it will be NAD+. It's really like a reverse of oxidative phosphorylation. Here NAD+ will accept the electron and will become NADPH. Remember, so far, in oxidative phosphorylation, we always say this, right? Gain an electron, then become NADH, right? Yeah. The plant version is you're going to have P inside. Okay. Think of them all together. I have not talked about the last part, right? Okay, as electrons flow down the electron transport chain, the usual will happen. Energy that's released will be used to pump protons from the, in this case, the stroma to the thylakoid space. So this arrow represents the flow of protons pumping in. And as a result, we end up setting up what we call a proton gradient. Very high on the inside, very low on the outside. And that means all the protons will want to flow out of ATP synthase. And as a result, we will produce the usual stuff. ATP from ADP and phosphate. Yes. Oh, they've always been there. Yeah, in the stroma, inside the thylakoid space, just present there. Okay, usually the best answer to this is that you have to eat food, so it will come somewhere from your food. Yes. Yes. It's all simultaneous. Unfortunately, I don't have so many hands to demonstrate everything together. And that's why the video was trying to show all of it happening together. Yes, question? So you got questions here? Yes. Why are they coming down? Do you remember oxidative phosphorylation, when electrons flow down the chain? Energy is released and the proteins use the protons, pump the protons out into the intermembrane space. Yeah? So it's the same concept here, except that it's using the energy to pump the protons from outside in, like in oxidative phosphorylation. Are you asking why it's doing it? Oh, why it's doing it? So that we can set up a proton gradient, so that the protons will flow down the gradient back into the stroma, so that ATP will work. Yeah, to make ATP work. Yes. Say again. ATP synthase, is it? For your information, the active site is over here. Yes. Okay, so I think you are not very happy that how come protons are just here? Is it? Okay, I understand. Okay, I think it's a misconception to think that in oxidative phosphorylation, all of the H plus came from NADH. NADH only contributed just a little bit more of H plus. But pre-existing inside the intermembrane space, there were already protons. Inside the matrix, there were already protons. Where did they come from? Okay, they came from all the foods we eat. When we eat foods, there are acids in our food. It will give off hydrogen ions. So, in this case, it seems weird. We don't have anything that can help to give off H plus. But actually, they already pre-existing because we do consume foods that contain ions. Or plants don't eat, but plants do absorb nutrients from the soil up into the relevant parts of the plant. Yeah. Remember, they say that plants absorb mineral salts, all these kind of things. Yeah. Also will come with a lot of ions. Okay, anything else? Because after this we'll try to compare and contrast. Huh? Any other questions? Yeah. Yeah. Well, it's very hard to teach a lesson to a student person. Yes. 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 Water, yes. Okay, good job. Alright. So better? Okay. Okay, now the fact that this chain looks so similar to the one in cellular respiration, it will be very natural that I'll ask you to compare and contrast the two. So let me help you out with that right now. Let's compare and contrast as many things possible. Number one, okay, first point of comparison. Okay, who is the electron donor in I'm going to say light dependent stage? Who is the electron donor? Number two, who is the electron donor in oxidative phosphorylation? Okay, that's the first thing we can compare. Now, I won't tell you what it is, you figure out yourself. But if I tell you what it is, I'm really telling you what was the essay question. Or whatever. Number two point of comparison. Can you tell me who is the final electron acceptor in the chain? Who is the final electron acceptor in the light dependent stage? Who is the final electron acceptor in oxidative phosphorylation? Second point of comparison. Rather difference number one, difference number two. Although I'm just asking you these questions, if you can fill it in, can you please try? Then you are comparing on the go. You could compare it in a table form if you want to. Okay, so 2 points of comparison. You can compare electron donor. Who gives the electrons at the start of the chain? Who receives the electron right at the end? Feel free to discuss with your friend who you think are the donors, who you think are the acceptors. State. Okay, third point of comparison, I'd like you to try. The electrons are pumped from Okay, in the light dependent stage, where am I pumping from? From where to where? In oxidative phosphorylation, I'm pumping from where to where? It's not sufficient to say pump out or pump in. Out or in is relative. So pump from where to where? Okay, at the very least, you should be able to fill in the blank for light dependent stage. Tyloid stage. Point of comparison number 4. The electron transport chain is found on which membrane? Okay, for light dependent stage, where do you find the electron transport chain? For oxidative phosphorylation, where do you find the electron transport chain? Once again, I will not tell you the answers. I can only share with you the points of comparison. But if you work together, this shouldn't be difficult to fill in out. Comparison number 5. Does the electron transport chain require light or slash chlorophyll or slash photosystem? Okay, so it's not yes or no. Okay, so light dependent stage, will you require the 3? Oxidative phosphorylation, will you require the 3? Oh, oops. Just realized it's another point of comparison that I forgot to tell the others. Oh well. Yeah, for the system. Okay, last comparison. What are the products? Right, this is all about interactions. What are the products of the light dependent stage? And what are the products of the oxidative phosphorylation? Okay, this one I'll give you a clue. For light dependent stage, one of the products is ATP. Okay, is there anything else? Okay, this one let's do it together. You might say oxygen is also one of the products. Any more? Okay, thank you, and NADPH. Okay, so what's the products of oxidative phosphorylation? Okay, so we have just listed down 6 points of contrasting features between the two electron transport chains. Okay, we'll take a short break. Yes. Yeah. Okay, we'll take a short break. You can bounce with your friends what you think the answers might be, but I will not share the answers with you. someone alphabet. Thanks, Megan. NADP. Thank you. I see something wrong here, you know? Yeah, it's protons, huh? I hear your discussion here, I'm very confused. Something's wrong here, it's not electrons. Okay, do not pump electrons to pump protons. Oops, I wonder if I destroyed the previous one. If you discuss with your friends what you think the points are, I tell you everything there, I will tell you the answer here. No. Okay, but with every compare and contrast, you don't just need differences, you need similarities. Okay, the similarity I'm going to tell you up front, if you can tell me. Okay, so what are some similarities between this and oxidative phosphorylation? Both involves proton gradient. So similarity 1, proton gradient. In both cases, we need to set up a proton gradient. Any other similarities? Any obvious similarities? Okay, similarity number 2. Both of them have ATP synthase. And therefore, any other similarities? Similarity 3. Both produce ATP. Any other similarities? Sorry? Both need water. Okay, let me think. Is that true? Okay, in light dependent stage, it needs water at the start. For oxidative phosphorylation, it produces water. Yeah, so it's a bit different. Okay, I think I can only spot 3 similarities. Yes, you see got another similarity. Okay, so you see says perhaps a fourth similarity is that protons, okay, let me put number 4 with here. Similarity 4. Electrons, is it? Electrons are transferred across electron transport chain. Okay, fair enough, there's a similarity. Okay, I think that's about all I can provide. Okay, the blanks you fill in yourself. Because that will really force you to draw similarities and study the previous chapter. Yes? Yeah. You mean which memory? Okay, we'll take a short break for you to consolidate your learning. Okay, you can read this page again to see if it makes more sense. Yes. Oh, so we will go comparison by we will not describe everything in one picture describe. Then next we will it's like I'm pumping water all the way up, right? Yes. It's okay. You take a start. The question is, what is electron donor at the start? It's not asking at the start. But you're right, equals is the word. Okay. Okay, then we're gonna come to a close to this chapter. If you are ready, we are going to conclude with the second part of photosynthesis. Good news is that photosynthesis only has two stages. Okay? I want you to look at the second stage. Does this stage look familiar to you? Does it look like any other stage in oxidative phosphorylation, in cellular respiration? Krebs cycle. Does it make sense now? Remember? Right after Krebs cycle was oxidative phosphorylation. Now we are going backwards. Yeah? So after we are complete with the electron transport chain, we have a cycle of our own right after. Can you see it's really like a reverse? Except, remember this, in the Krebs cycle, the aim was to pull up as much electrons possible, and also from there we release a lot of carbon dioxide. here, we do the reverse. We will take we will instead channel all of the NADPH, all of the ATP to the next stage, the light independent stage. At this point, we will pump it into a cycle of reactions. Yeah? What's the point of NADPH? Well, if we supply NADPH, it's just like what we did by the reverse now. Now we get NADP plus, but we will now, electrons will now be given to the reaction. ATP is also needed in this process. ATP, then you end up getting ADP. But the output is sugar. So I'm missing one more thing. We need carbon dioxide. So if I want to put it very simply, I think of it as a reverse Krebs cycle. In Krebs cycle, we took out carbon dioxide, we took out electrons, we also, did we produce ATP? We produce ATP. In this cycle, we use ATP, the NADPH will supply electrons, we will also use CO2. And the end result is that we produce a sugar. Ryan? Okay, so I haven't elaborated yet, right? Sorry, Ryan, you have a question? Okay. If you read your textbook, you'll find that this sugar is, they say it's a 3-carbon sugar. Yeah? Does it say in your textbook? What is produced is a 3-carbon sugar. Do I need to really go to that page? Yeah, you can see a 3-carbon sugar. I'm not straight away from the idea that we're using at start. Interactions. All we need to know is what is involved, what is produced. Yeah? And we're doing the same thing here. What is involved? NADPH, ATP, CO2. What is produced? NADP plus, ADP, and sugar. Yes? Yeah, this is NADPH. Oh, sorry, what did you say? NADP, forget it, D. Like that? NADP plus. Yes? Oh yeah, it should release a phosphate group to get NADP. So I should. So if you ever forget this particular cycle, just recall Krebs cycle, reverse up. Okay, this cycle also got names. That one is Krebs, this is Kelvin's. Huh? Do you say Hobbes cycle? Kelvin and Hobbes. Yes. Kelvin cycle. You don't Google. Why you interested? Oh. I don't know, I don't think I can. Okay, notice we call this particular stage the light independent stage or the Kelvin cycle. Okay, by right, this you all people asking what it is. Okay, Kelvin cycle. That's because this cycle does not require light. Only this part requires light, right? So you know what this means? That technically if I want to grow a plant, I could supply it with light for a while, and if I were to switch off the light, photosynthesis can still run, at least the second part of it. Yeah? So plants don't die immediately when you switch off the light. They would have stored up, or they would have produced enough ATP, NADPH for this second part, and they can conduct that in the dark. Yes? Why humans 1 minute without oxygen we die? I think it's because human beings were very big, and so we do require a lot more ATP. Yes? What about trees? Trees are very big also. Okay, then maybe I ask you this question instead. Sorry? Yeah. But you look at a tree, right? They can afford to one leaf die, just drop it off. But very different. It's like one with my arm. It's not like you can just let one arm go, right? And then just not enough oxygen. It's not like I hold my breath, right? Then the longer I hold, right, one arm drop off, then the arm drop off, right? Okay, what do you think about this? Okay, this is a very interesting question. You see, the point of cellular respiration is so that we can produce ATP, right? What is the point of photosynthesis? I ask you, what's the point of photosynthesis? Produce food, right? Okay. Then my next question to you is this. What is this food for? Okay, this food, plants also carry out cellular respiration? Yeah, this food will be used for cellular respiration in the plant itself to then produce what? To produce ATP. So my question to you is, why is the plant producing sugar so that it can produce ATP again when actually at this point it really produces ATP? Sorry? No, so my question is, why don't we just stop it here? Why must I channel this ATP to produce sugar when the plant can actually just produce ATP itself? Okay, so you just answered the question. You can store sugar, right? You cannot store ATP actually. Sugar can be stored. That's what makes plants amazing. They can store the food they make. Secondly, plants, there's no perpetual sunlight, right? So if plants only had the light dependent stage and does not have this stage, that means to say the moment it is dark, the plant will die, right? But so what are plants doing really? They are making use of daytime to produce as much sugar so that come night time, they can use it also. Yeah? Sorry, that was just an interesting question raised by a student. What's the point of going through all this when you can already produce so ATP? Yeah? Yeah? Okay, I want to say at night, daytime do plants respire? Yeah, they do also. I think you all learned in primary school, right? So the sugar is being used in the day and at night also. I would say at night it really has to dig into its reserves. Yes. Okay, I said SARS, he observes that there are some trees that will shed its leaves, right, come winter time. How come it can still survive? So, like the bears that go for hibernation, the plants can also store as much nutrients as possible within its bark. The parts that are not photosynthesizing, the parts that come the winter time when there's not as much light, it could still tap into the reserves that it's been storing up. So I would say those trees that survive during winter time without much sunlight is tapping into the reserves just to carry out cellular respiration to live. Then when there's more sunlight, it starts growing out its leaves again to tap onto the light to produce more food. Yeah. Oh, is it possible rather than tapping into the reserves of the sugar, why not I just slow down all my processes? Actually, it's possible. And you also learn that when it's lower temperature, enzymes already don't function as fast. So actually, it will slow down come winter time. Yes. What do you say? Do obese people take longer to start? Yes. Yeah, I've never asked myself this question before.
Yes. I think it does.
And what basis are you? What basis are you asking this question on? Yeah, I don't know leh. Yeah, I would say that it's not that if I have more fats, then I will take longer to die. Like it cannot. I don't think so that if I have more fats, you go and lock me out of the room, I can survive in there for longer time. Okay, because at the end of the day, there are daily processes that require energy. They require energy to be released quite fast. And I think when you store things as fat, right, the conversion to get the energy back isn't as fast. Yeah. You need the energy for what? You need the energy to make fats. Oh yeah. You will definitely need some energy to do the conversion from fats back to energy also. So I do think that even whether you have a lot of fats or not, doesn't mean that you will survive longer.
I know it does.
But if we have two people, one with a lot of stored fats, another one with very little, should the one with less fat eat the one with more fats?
That's a question for
Ethics. What? Is there consent? I'm really sorry, but is it okay if I take a bite out of the bicep? Yeah, I read somewhere right there. Some countries, right, when they fish for crabs, right, they don't take the entire crab. They just take one claw away.
Yeah.
Because they can regrow it, right? Then like that you don't have to kill the crab. Then they just take one claw away, then you go back. Then when you grow already, you just take the claw away again. After all, people eat crab, I also eat the fleshiest part, right? Okay, to end off the lesson or the chapter for this term, can I get you to go to these two graphs? You'll very likely see these two graphs pop up very often. I just want to help you understand what these graphs are trying to depict. Let's look at the axis. On the left-hand side, the top graph says absorption of light by chloroplast pigments. On the bottom, the x-axis, you see different wavelengths of light. Each wavelength corresponds to color. So at 700 nanometers of wavelength, that it represents red color. This spectrum, what color is that? Is that purple? I don't know what color that is. Pink? Okay, so what this graph is trying to show is, they show you three different pigments, A, B, and carotenoids. These are just different versions of chlorophyll that exist in plants. If I were to ask you, which wavelength of light does chlorophyll A absorb best at? What would you say? Chlorophyll A, 430? If I ask you, for chlorophyll B, and which wavelength does it absorb light best at? You would say? 470, 460? Okay, so this is the only skill I need to pick up to be able to interpret the graph in this way. Notice that, therefore, if for chlorophyll A and B, actually they have another wavelength of light they are quite happy with towards this end. So plants, right, they actually really like bluish light and red light. Yeah? You know what light they don't really find useful? Green, right in the middle. And that's why plants look green, because that's the light they just don't want to take in. Yeah? What does that help? You have a question? Yeah. Okay. How come we see some leaves, right? Look, orangey color. Right? I believe there are two reasons why. One is there are some oxidation processes beginning to take place. Another, there are some chlorophyll pigments that look that color also, like orangey, reddish color. Next, yes.
So does that mean some plants with purple color leaves will like purple color?
Okay, if a plant is purple color, does it mean it's absorbing purple light? Okay, that means actually.
Entire leaf is purple and entire plant.
Yeah, that means right, it's actually not absorbing purple light, right? That means it's absorbing some other spectrum of light.
Yeah.
Yeah, some other spectrum of light. Well, some of you may be wondering this. Why do plants like these two spectrums and not green? Okay, there are many theories to this. Scientists believe, if you go back in the evolutionary tree, that the reason why plants are green is because there was another organism that was able to photosynthesize first. Yeah, and that's bacteria. So there were bacteria deep in the depths of the ocean. They were able to photosynthesize, except that they made use of green light. And these bacteria look purple. So plants came after these organisms. So to reduce the competition, they instead absorb the light. These bacteria were not absorbing, and therefore they look green. So this is one of the theories. This is one of the theories. Imagine if the bacteria and the plant both photosynthesize using the same spectrum, right? Oh yeah, what's that competition? I would suppose if you are better at one than another, you could overgrow, shade out the rest, you know. So there is competition. If you grow better, you will shade out the other. Okay, let's interpret the second graph. This says rate of photosynthesis measured by O2 release. Do you see a relationship between the top and bottom graph? Yeah? Yeah, what conclusion can you draw from the top and bottom graph combined together? Yeah, as Rachel put it, the more light is absorbed, the more photosynthesis occurs. Make sense? Yes. Except that this wasn't that obvious to scientists in the past. Needed to do these two experiments to be damn sure that yes, more light equals more photosynthesis. Yeah? So if you ever see these two graphs, don't be intimidated. Just read the axis. That's all they want you to interpret. If I were to ask you, at which wavelength of light is it the best for photosynthesis, you would tell me? Like over here, not bad, right? Yeah. If you ever go to the agri-tech, go inside the containers. Inside the container, there's only purple light. Yeah? That's because don't waste energy. No point shining green light, it's a waste of energy. So inside the agri-tech, the hydroponic section, only shine purple light, red and blue. And it's very interesting. Because you think about it, if it's shining purple or red and blue light on the plants, right? And the plant is absorbing everything, nothing else is reflected, right? Okay, so these plants don't really look green under these lights. They look like a grayish color because everything's being absorbed. Yeah? So Megan asked this question last time. In complete darkness, is there still color? Because at the end of the day, color is simply a leftover of what's reflected off from the sun. So in complete darkness, we are all the same. We have all no color. Color is only a result of when there is light. So in darkness, there's no red, there's no yellow, there's nothing. It's not that we cannot see color, it's just that there isn't color. The natural state is that there is no color. Wow. Okay, we have come to the end of term 1. Wow. Wow, time flies, huh? Right? 10 weeks past. Okay, so we have covered a few chapters by now, right? Okay, in term 1, this is what we covered. Number 1, I don't know if you remember. Number 1 is,
Cell cycle.
is it cell cycle? I cannot remember.
Yeah.
Cell cycle. Okay, what was after cell cycle? Okay, gene expression. Then we went straight into these topics already. I suggest don't study them separate, they are very similar. So study them together. It is after one chapter together, energetics. Because we are studying how energy is made, how it is used. Okay, that's why we call it energetics. Mutation is under this one over here. So we have 3 chapters that we are going to assess you on. See how much you understand when you come back from the March holidays. I'd like to emphasize that we will use this quiz not so much to stress you, but so that you can really put your notes to the test. Okay? It's a very rare opportunity. You don't know under time conditions whether your notes are useful or not. Okay? So let's try it out after the March holidays. Spend the time making good notes. So that come your mid-years, you will have 3 less notes to make. Okay? Okay, so by the end of March holidays, you submit a scan for me online so that I can keep track. Yeah? Okay. Okay, I'd like to preempt you. So we have cellular respiration, TE. There will be another T coming out, photosynthesis. But it's very rushed to complete all this weekend. Right? Okay, so that one by the end of March holidays complete. But then that will also mean that I won't have time to give you back any feedback in time. So I'll record a video earlier so that after you do, you can watch immediately, you can make your notes. Okay? So for the photosynthesis TE is the one where I don't think I can give feedback in time. Yeah? So photosynthesis TE. Okay, I think our class is in a little bit of a pickle because sound is not playing. Okay, so to help your class out, can I send you the Zoom link for later? Where is the Zoom link? Okay, in the event later your PM comes in and the sound still doesn't work, can you access the Zoom link that I'm about to send into this chat? Okay, I'll send to the core 3 biology. There are some who are not in this chat for biology, so maybe you have to send to each other. You received it? You got it? Yeah? Okay, you got it? Yeah. Okay, so please be nice to our sharer. I've even asked the sharer to provide his product. So I've invited someone who went through a lot of prototyping to create a product that he wants to use to help society. He and his team created edible cutlery that is packed with nutrients. So that when you eat, after you finish eating, don't throw away, just eat it all together. So that's his product. And we wanted to invite him to share his prototyping journey. They're very nice. They are going to give each one of you one of their products. Yeah? So later on, you can look forward to them. So these opportunities aren't easy to get. I hope that you all treasure these opportunities. So there will be a few flavors. It will be a draw of the luck. What kind of flavors you get? Okay, there's banana goji, there is caramel, there is strawberry. Okay, so you have any questions for them on how they arrive at their prototype and product? Now's the great time. Yeah? I'll see you over Zoom.
Alright, I will make sure to be high.
And actually their project started off at university, started off in school. So a lot of projects starting off from school can really branch out to new opportunities. Hey shucks, I haven't taken this now, oh my god. Okay, but that's all for today. Hey, thank you so much. Who's your PM?